Hello, everyone, and welcome to today's webinar. My name is Mingjuan, and the Director of Marketing at uBriGene, and I will be your moderator today. I see some old friends from our previous webinars. Welcome. iPSC reprogramming webinar, high efficiency iPSC reprogramming for regenerative medicine, RNA-LNP complete kits. This webinar is a part of our series on advanced therapies, development, and manufacturing. If you missed our earlier sessions on CAR-T, lentiviral vector, RNA-LNP, and CRISPR, we had a few, and also we had an iPSC webinar this March. So you can watch those recordings, and we post on our website. So now it is my pleasure to introduce the webinar speaker today, Dr. Scott Patterson. Scott is a PhD-trained molecular and cellular biologist, as well as a seasoned business development leader with over 17 years of experience spanning both biotech R&D and commercial strategy. He has played a key role in advancing therapeutic discovery and development across various modalities, including biologics, cell and gene therapies, and genetic medicines. Scott has extensive expertise in the iPSC space, including reprogramming, engineering, and translational research for regenerative medicine. Today, he will discuss high-efficiency iPSC reprogramming from PBMCs using our innovative RNA-LNP technology and share exciting data on the differentiation into neurons. Hey, Scott, without further ado, I'll hand it over to you. Thank you, Scott.
Thank you very much, Mingjuan. I am going to turn off my camera and just focus on the slides here. So let's go ahead and get started. Thanks for that introduction, Mingjuan. Much appreciated. So we've already done our introductions, and we'll jump right into the uBriGene iPSC portfolio updates, and hopefully we'll have plenty of time at the end for Q&A. And like Mingjuan said, feel free to submit your questions as they pop into your head, but we won't address them until the end of the presentation. So this is kind of going to be our template slide as we move through the presentation. If you were part of our March webinar, you also saw this slide, but it seems to work pretty well. And it basically just shows the workflow of sourcing donor cells, reprogramming, doing editing, which we won't talk much about today. We talked quite a bit about that in previous webinars. Banking, and then new updates on our ability to do differentiation specifically for neuronal lineage cells. And so we like to set the stage for our webinars by starting with some of the key challenges that folks, in this case in the iPSC space, face. And the first of those is donor cell access. It's just a very complicated, complex process. There's a lot of logistics that go into it. And so we have taken all of those headaches away from you. We have a very robust FDA compliant sourcing process in place. And we also now are offering off-the-shelf GMP iPSC lines, so you don't even have to worry about sourcing. You can jumpstart your programs with already GMP compliant iPSC lines. So that's a really nice offering that we now can provide to our clients. Safety and genomic instability is another hazard with classical approaches that utilize viruses and transfection for doing iPSC reprogramming. Our RNA-LNP cocktail, as we'll talk about in a few minutes, is integration free, and so very, very low, if almost virtually zero genomic instability risk, I should say. Reprogramming efficiency is always a bottleneck when generating and generating iPSCs, screening for clones, characterizing clones, and then moving on to differentiation. And I hope that you'll appreciate when you see the data today that we make this step of the process, which used to be probably the most laborious step of all, a very efficient step in the process. And then lastly, we're really excited for this latest update. We now have decided to commit resources to working out differentiation workflows. And so we're really excited to share with you some of the processes that we've developed for neuronal, like Ming-Juan said, for neuronal lineage cells. But we're also working on other cell types too. And during the Q&A, we'd love to get your feedback on which clinically relevant iPSC-derived lines, or I should say cell types, you're most interested in accessing. And suffice it to say, we have one of the big bottlenecks with iPSC workflows is you have to go to one place for donor sourcing, another place for genome editing, another place for banking, and then somebody else for differentiation. And to be able to find that all in one reliable supply partner is really, really valuable. And that's what we're trying to provide both to researchers in the iPSC space, but also for those using iPSCs to develop cell therapy programs. And so this is a nice slide that, again, if you joined our March webinar, you saw a version of this back then, but it's a nice way to describe our offering. Starting in the upper left-hand corner with that O1 hexagon around FDA-compliant donor sourcing, which we talked about at length in the March webinar. I just have one slide to remind you or to introduce you, if you weren't in the March webinar, to what we can do with regard to making donor sourcing very efficient. We're going to spend time, and you can see from the title of the webinar, most of what we're able to do in the iPSC generation and editing banking space is built on the foundation of our RNA-LNP reprogramming cocktail. We developed this internally to make our GMP manufacturing and iPSC banking services more efficient. And we realized, well, why don't we offer this as a product so that more folks than just uBriGene can benefit from it? And so we're really excited about the commercialization of this as a kit and some of the early results, which we'll show you during the webinar, not just uBriGene results, but also some of our key collaborators' results. iPSC banking is something that we do a lot of. And one of the commitments that we made in the red box in the lower left-hand corner is that we're going to offer GMP iPSC lines off the shelf to folks. And we have our first one of those lines available now, and we'll continue to add to that catalog of iPSC lines, GMP iPSC lines that we can offer to groups in the iPSC-derived cell therapy development space, which enables you to start your program at 0.3 here on the slide as opposed to 0.1, which basically cuts months off of your program timelines. The lower right-hand corner, I'm not going to talk a lot about today. We talked a lot about that in previous webinars, both in December of 24 and March of this year. But suffice it to say that if you need gene editing solutions, either research grade or GMP grade, we have those available. We have great platforms for offering the gene editing nucleases, either in an RNA or protein format, for offering platforms for delivering knock-in payloads, either non-viral approaches or AAV-based approaches. And then we also have a novel platform for generating guide RNA that we talked about at length previously. And if you're interested in those gene editing solutions and our IVT guide RNA process and products, reach out to us and we can set up a separate discussion. And then lastly, one of the new innovations that we wanted to introduce today on the webinar is we now have developed some differentiation workflows. I'm specifically going to show you some of the results on some neuronal lineages, but we're also working on immune cell types as well as muscle cell types. And so we're really excited about highlighting the things in the red boxes on this slide today. And so first of all, just a quick refresher about Oh, I'm sorry. We're seeing for a conference I'm going to next week, seeing updates on partnering. In terms of donor sourcing, so like I mentioned earlier, this can be a very, very complex process. And if you've never done it, it's even more daunting. And so basically, we have put in place a very, very efficient, fully FDA compliant process, working with the medical clinics to recruit the right facilities and the right clinical medical centers and the right PIs at those medical centers. To source the materials, we have CLIA certified testing labs in place for all of the testing that's required in order to evaluate the donor materials as well as the lines that are generated from those materials. And then we have a network of couriers that have tremendous expertise and tremendous global reach for providing door-to-door supply chain courier services. So that's a really nice benefit to be able to have if you're thinking about taking your iPSC innovations to the clinic, to have all those FDA compliant processes in place. And that's all I'm going to say about that. But if you have further questions, happy to set up a separate call around what we can do to help you in that regard. So let's go ahead and jump into reprogramming. That's what we're really excited to talk about today. For those of you who don't know already, we have commercialized, we used it for years internally, and we decided to commercialize an iPSC reprogramming cocktail that's RNA-LNP based. And so basically we've identified the key reprogramming factors that we find work best for iPSC reprogramming from either fibroblasts or PBMCs. And we've pre-built an RNA-LNP formulation with those reprogramming factors. Because it's RNA, the first bullet here is it has no genotoxicity risk. There's no chance of integration. I mean, we obviously still do karyotyping just to confirm that, but there's basically zero risk of integration and genotoxicity. As I mentioned, it's in a pre-formulated, ready-to-use LNP mix, and we're getting very positive feedback from groups that say, indeed, it's very easy to use, and they're getting very high iPSC reprogramming efficiencies, as shown in the third bullet. For fibroblasts, we typically see, single digit to 10% reprogramming efficiencies. So that means, typical reprogramming workflow, you start with 100,000 donor cells, you're going to get thousands of colonies showing up in about 10 to 15 days. For PBMCs, as many of you know, this is a more challenging process, but we have our newest version of the cocktail, which we'll talk about in a little more detail, gives around 0.3, very specific here, 0.31% reprogramming efficiencies. And basically what that means is if you start with 100,000 PBMCs, you're going to get hundreds of colonies showing up in 10 to 15 days that you can work with. And what that means, being able to generate high quality colonies right out of the gate within 10 days, it dramatically reduces all of the effort and all of the money and all of the time that goes into lengthy clone screening campaigns. You get to useful clones that you can pick, expand, characterize, and start working to differentiate much quicker than classical approaches. That I think we have a slide that talks about some of the classical approaches. And it's a very flexible system. We've gotten feedback from groups that it works for feeder-based iPSC culturing as well as feeder-free culturing. So this kind of compares our RNA-LNP reprogramming kit to some of the classic approaches. So first, I'll just compare it to RNA-based transfection approaches. And as you look at this table, basically row 1, row 3, and the last row are really the classic improvements that we've made here. So we have much higher reprogramming efficiency with our RNA-LNP approach. We don't require, cytotoxic transfection electroporation, which beats up the cells quite a bit, which you don't want with primary patient-derived cells. And then lastly, compared to RNA used in RNA transfection approaches, our RNA-LNP formulation is quite stable. We now have stability studies out past a year, and it shows very robust performance after a year of being frozen. And then some of the more classic approaches for iPSC reprogramming, sendivirus and, naked DNA episomal vector approaches. The key differentiator with regard to sendivirus is that next to last one. So, viral persistence is a huge, huge issue with sendivirus. It's around for many, many generations as you passage the cells. And so that poses a carcinogenicity risk. And so a safety risk that you have to deal with and be very concerned about. And obviously our non-integrating RNA-LNP formulation, that's basically zero risk with regard to that. And then with regard to naked DNA, episomal vectors, those required transfection, either chemical transfection or electroporation payload delivery. So that's very cytotoxic on the cells. The efficiency and the amount of time that you have to wait for colonies to form is much longer with our RNA-LNP approach. And those are the big ones with regard to naked episomal DNA viral vector approaches. So suffice it to say, our approach, our commercial product is very, very attractive, and we're hearing that not only from our laboratory team, but from some of our early clients, and we'll show some early client data. So what have we done? We introduced this RNA-LNP cocktail in March for the first time in a webinar. What have we done since then? So really the innovation priorities have focused on three major things. One, first of all, is RNA optimization. So we've done a lot of work around optimizing the specific RNAs that go into the reprogramming mix. We also collaborate. We are not a lipid team internally, but we've set up some strategic collaborations with groups who do lipids and LNP formulation extremely well. And so we've screened many, many more lipids since March, and we've identified some ideal lipids for the LNP formulation. And then we've also developed a proprietary enhancer B. Sorry, it has to be so mysterious, but it's still proprietary and patent pending at this point. So suffice it to say that we've identified some additional chemicals that further enhance our reprogramming efficiencies. And so those three innovation vectors have led to being able to treat the donor cells, be they fibroblasts or PBMCs, less frequently. And so that means, less stress on the cells, and with smaller volumes as well compared to our previous protocols. And so much gentler process and a much improved reprogramming efficiency, especially with PBMCs and difficult to reprogram cells like, patient-derived disease tissue cells and things like that. So we've gotten very positive feedback on these latest innovations and how they're translating into better, more gentle, more efficient iPSC reprogramming for challenging cell types like PBMCs. So here's what the workflow looks like. You saw this workflow when we presented our webinar back in March, and that was for fibroblasts. We typically treat those cells daily for 8 or 9 days, and we see very high reprogramming efficiencies from those easy to reprogram cells in the 4 to 10% range, like I said. The new innovations that have been introduced into the iPSC reprogramming, our iPSC reprogramming kit, now make it much easier and gentler on the PBMCs. This was the main reason that we made these innovations, was for groups who are working with PBMC-derived lines. You only have to treat the cells every other day for about 6 days. So fewer treatments mean less stress on the cells. And we're seeing colonies show up in the same time frame that we saw with our fibroblast focused kit in about 10 to 15 days. And we'll show you data not just from us, but from some of our key collaborators confirming that. And so you get about 0.3, there's that 0.31% efficiency again. But basically you're going to get from 100,000 starting PBMCs, you're going to get hundreds of colonies to work with. And so that's very, that's great. That enables you to hit the ground running and get useful colonies very quickly. And that's about 100-fold higher than most of the RNA products that you're going to find on the market today. A little bit about our certificate of analysis. So this is the typical QC panel that our RNA-LNPs go through for our reprogramming kit, and also for RNA-LNPs that we make for therapeutic development partners. So I won't go through all of the details of this, but obviously we look at appearance, we look at the biochemical performance, and then we also look at the reprogramming efficiency before releasing the product out to our clients. And we can have a much more lengthy discussion about just the RNA QC that goes into the RNA that gets incorporated into the reprogramming cocktail, or about this panel that I'm showing in this slide. So data from our uBriGene team shown here. So this is with fibroblasts. We showed this on the previous webinar. We start to see colonies around 9 or 10 days. And most of the time we pick our colonies at around day 15, and they give very nice results in terms of expansion and characterization and subsequent differentiation. This is some new data. So this is our PBMC reprogramming, and similar to fibroblasts, we start seeing the appearance of colonies around day 10 to day 12. And usually we pick our colonies in our PBMC workflow around day 15, like I showed for the fibroblasts. So we're getting really robust and reproducible, high quality results with this approach. Have a couple of key collaborators. Oh, that's not the next slide. Sorry, I jumped the gun. And then obviously we do the classic QC on any iPSC lines that we generate, looking at stemness of our iPSCs, looking at pluripotency and embryoid body differentiation studies, and then G-banding karyotyping to ensure that we're seeing genomic integrity and there's no genome instability issues. And so the classic markers, nestin for the intermediate filament, and MAP2, the microtubule associated protein for ectodermal differentiation, alpha fetoprotein for endodermal, and then SMA, spinal muscular atrophy markers for pluripotency. Now I get to show some client data. So we're excited in this webinar to be able to share some data that some of our key collaborators have given us permission to share. So this is an academic iPSC core that has utilized our latest version of the mRNA, the RNA-LNP reprogramming cocktail with PBMCs, and they let us know that they actually saw and picked very high quality iPSC colonies at day 12. So like I said, but usually we do it at day 15, but they actually picked the colonies at day 12. They're expanding and characterizing those colonies, so we don't have all of the typical QC that goes into characterizing the colonies, but suffice it to say that this core has been very pleased with the performance of the RNA-LNP reprogramming kit for PBMCs to date, and plans on using it more regularly moving forward. So that's our update on the iPSC reprogramming kit innovations. And I want to segue in, I'm skipping over the gene editing. We talked a lot about that in the past, but again, if you have questions about that and would like to learn more, obviously you can visit our webinar page on our website, like Ming-Juan referenced, and view the webinars, but we're happy to set up a call and talk about those solutions. But let me go ahead and move into our iPSC banking capabilities. This is a very, very common request from our clients. And so we do this a lot. So obviously I already told you on one of the opening slides about our FDA compliant donor sourcing, which streamlines that process. I've told you about our RNA-LNP reprogramming cocktail, which leads to the generation of iPSC colonies at a very high efficiency, with fibroblasts or with PBMCs. And then obviously we have the ability to isolate lines for research purposes, for those who are working in the research space, building disease models or looking at disease mechanism of action. or doing drug screening. But we also are very expert at single cell cloning and deriving GMP grade iPSC lines and iPSC master cell banks. And we'll show you some data from one of our early GMP iPSC master cell banks from one of our clients. This is the QC panel that we typically carry out for the parental cells for iPSC seed banks, and then ultimately for our iPSC master cell banks. I won't go into all the details, but you can see, the usual safety and sterility markers. We look at markers of genomic stability, obviously. For GMP banks, we need to get those out for adventitious agent testing, and so that gets done for GMP banks. And then obviously we look at pluripotency markers and stem cell markers through flow, through teratoma formation assays, and through embryoid body formation assays. So this is our second slide from a strategic collaborator who agreed to let us share the data. And that is, they took one of our early iPSC master cell banks, our GMP iPSC master cell banks, and looked at it for stemness, for proliferation. And they report back that they're very pleased with the way the cells are performing in terms of just maintenance. So this is showing a couple different media that they wanted to look at. These are commercially available media that they kind of use as benchmark media and that we also use internally, showing good cell yields, viable cell yields, and showing good population doubling times. Actually quite impressive. They said most of the lines they look at are typically, more like 24-hour doubling times. Ours are showing around 20 to 21-hour doubling times in the different media, and our internal team sees that as well. In terms of pluripotency, obviously they stained them for some of the classic pluripotency markers, and those all generated 98% plus percent marker positivity in the lines in both media conditions. And then they also sent them out for genomic stability analysis, and those came back completely clean, and with the third-party outsourcing partner, which they used for their genomic stability analysis. So we were thrilled, and they're excited. They're going to continue to use this line as well as subsequent lines that we developed for them in their workflows. And so with regard to iPSC banks, we are able to, as I've said multiple times, source donor material if you need for us to do that in an FDA compliant, very efficient way. So they take that headache away from you. Once we get cells in, we reprogram them with our very efficient, low genotoxicity risk RNA-LNP reprogramming product. And happy to say, and you saw donor data for iPSC reprogramming as well as that characterization of our GMP MCB banks, that they've been very, very rigorously characterized and tested, not just internally, but with our external partners and clients. And we can offer these products and services at a research grade environment, or we can do these in a GMP grade environment for those who are wanting to take their iPSC innovations to the clinic. And then the last piece, so we've talked about donor sourcing, we've talked about reprogramming, we've talked about banking, and then we're going to talk about some neuronal differentiation workflows that we're developing. But before I jump into that, I believe Ming-Juan has a few poll questions. Do I need to stop sharing, Ming-Juan, for you to do that?
No, you don't.
I don't. Okay.
Yeah.
Thank you.
So I have a few poll questions. So this is the first poll. I will give you like 10 to 15 seconds. So to give you another few seconds. All right, thank you. This is the second Poll question. Give you another few seconds. Thank you. And this is the third poll. Give you a few more seconds. Thank you for answering those polls. Scott, you can continue.
Are you able to share the results easily or no? I'm just curious to see what they look like. Okay, no worries. Thank you for taking the polls, guys. Let me jump back into my presentation. All right. So I'm just going to show some very preliminary data. Mingjuan made it out to sound like I'm an iPSC expert. I know just enough to know what markers need to show up when with different differentiation. But if you want to have a more technical discussion around these, the development of these workflows, we can get an NDA in place and have a call with some of our subject matter experts. The first one I wanted to show you is motor neuron differentiation workflow development. So, this goes from iPSCs through neural progenitor cells to immature motor neurons to mature motor neurons. The panels that we're showing here are with motor neuron progenitors, which is, I think, somewhere between an NPC and an immature motor neuron. And what you're seeing is the appearance of the oligodendrocyte 2 transcription factor and the PAC6 neuroectoderm marker showing up in the progenitors. And then on the far right, you see the merge of those markers. And then for mature motor neurons, you're seeing the islet 1 mature motor neuron transcription factor, and then the choline acetyltransferase, which is another classic marker for mature motor neurons, and then the merge of those two markers on the far right-hand side. So that's early work on our motor neuron differentiation workflows. We've also developed differentiation for GABA neurons. So this is about a 65-day process to get from iPSCs through neuroprogenitor cells into functional iPSC GABA neurons, the inhibitory cortical neurons that are used a lot in drug screening. And so this is showing, some of the classic markers for GABA neurons. The glutamic acid decarboxylase 67 is a common marker. And then we have TUB3, which is actually more of a pan-neuronal marker. And then we're showing the merge of those two markers with regard to the final differentiated product in this differentiation workflow. And lastly, we have some oligodendrocyte data to share. So this flows from iPSC to neuroprogenitor cell to oligodendrocyte progenitor cell to mature oligodendrocytes. And what you're seeing here is a merge of, the DAPI double-stranded DNA blue staining dye against PAC6 and nestin, which are neuroprogenitor cell markers. And then our old friend again, the oligo2 transcription factor, and then O4 is a surface marker for these oligodendrocyte progenitor cells. And so you're just seeing 4 different fields for each of these markers: oligo2 and DAPI, O4 and DAPI. And then as the cells mature and become actual mature oligodendrocytes, you'll see the appearance of myelin basic protein, here counterstained against DAPI in 4 different fields, and proteolipid protein again, counterstained against DAPI, 4 different fields. So we're making some progress on generating iPSC derived mature cell types that we think are important for disease modeling and for clinical development. And so I'm curious to see the results. That's why I was asking. I'm anxious to see the results of the poll questions to see if you guys are most interested in neurons or cardiomyocytes or immune cell types, whatever it may be. But these are the early results. And if you have other lineages, we would really, really value, because we're trying to figure out the right strategy on what cell types are going to be of greatest value to our clients. And so if you wouldn't mind providing feedback on that, feel free to reach out to any of us to say, hey, I could really use some GABA neurons, or I could really use some, T cells or NK cells or whatever iPSC-derived cell type you may need. That would be much appreciated to make sure that we're developing what is needed by our clients and partners. All right, so we're going to wrap things up. The 3 take-home messages that I hope you'll appreciate after the webinar is that we've put a lot of work into developing the latest version of our high-efficiency RNA-LNP iPSC reprogramming cocktail kits. We're getting a lot of positive feedback. You saw some third-party data that confirms the uBriGene data, which we're always very, very excited to see. The fact that we're now building out a portfolio of off-the-shelf GMP iPSC lines is already being well received by our iPSC cell therapy development partners, because that, like I said earlier, cuts months off their development timelines. And so we're excited to be able to bring that. And if, just like I said with the differentiated cell types, if you have specific needs where you need, I didn't mention, but the one that we now have that's available is from an Asian female, 38 years of age, and it's fibroblast derived. And so we want to make sure that subsequent lines we offer are from the types of sources that you need. So obviously we're going to make some PBMC derived lines, but if there's other specs that you need in terms of gender, in terms of ethnicity, in terms of, what sort of demographic they come from, or if you need iPSC lines from diseased patients, let us know that type of information, because that client feedback is what drives the development of these lines. And so we would really appreciate that. And then I just showed you a few slides highlighting the fact that we are now committing resources to developing iPSC differentiation workflows. And again, we would highly value your input on which differentiated cell types are of greatest value to you. And then as a show of our appreciation, we're offering access to our new RNA-LNP iPSC reprogramming by offering you a 50% discount on our 2-pack kit. And then we also have 6-pack kits available as well. And Mingjuan and our product management folks can fill you in on how you can process those orders. And so here's my personal contact information, email address and cell phone number. Feel free to reach out if you have any questions or comments. And at this time, I'm going to go ahead and stop sharing, and we can enter into the Q&A session. So thanks so much again, everybody, for joining today.
Thank you, Scott. That was a very insightful presentation. Yeah, as Scott mentioned, that we have time for questions. If you haven't submitted it in the Q&A box, please do so. There are already some questions came in. So one question is, we in our research lab testing, our reprogramming efficiency for PBMC is 0.31%. The question is this efficiency with the enhancer or not? Scott?
Yeah. So the answer is yes, Sakti. So that includes all of those 3 innovations that I highlighted in one of the slides. Optimized RNA, optimized lipids for the LNP, as well as the inclusion of that enhancer B. So that is what gives rise to the 0.3% reprogramming efficiencies with PBMCs.
Thank you, Scott. Another question is that transfection efficiency is very low for PBMC, and that is true. It's a primary cell, it's suspension, right? It's very low. This question said, is a combination of LNP plus electroporation if this can increase the efficiency? Scott?
Oh, that's an interesting question, Guanhan. That is typically not a combination that's used. LNP is usually used to get away from transfection, whether it's chemical transfection or electroporation. I don't even know if electroporation, I suspect, might damage the LNPs. So I don't think that's a viable approach. If any of you iPSC experts out there want to also address Guanhan's question, we have not done this internally, so I don't know that LNP plus electroporation would result in an increased efficiency. My gut reaction, having done a lot of transfections in my life back in the day, is that LNP and electroporation is probably not an ideal combination.
Thank you, Scott. Yeah, that's probably true. LNP, the purpose of formulating into LNP just for convenience, directly add to cells. And our enhancer will greatly facilitate the delivery to the PBMCs. Great. Another question says that what reprogramming factors are included in our LNP cocktail? If are there any microRNAs in the cocktail?
Yeah, so Wenley, we still have a patent pending for this RNA-LNP cocktail, so I'm sorry I can't share all those gory details with you. But right now, we're not in a position to be able to share. Hopefully, in a few months, we will be once the patent process moves along. But not able to answer that question today. So sorry about that, Wenley.
Oh, thank you. Another question said, why with the Sendai virus, you will need residual cleanup. You need to get rid of the virus.
Yeah, so Sendai virus, Guanhan, the reason people used it is because, it at the time that it came out, it really did increase the reprogramming efficiency. But the reason that it improves the reprogramming efficiency is that it persists. And so it sticks around in the cell, which is great for being able to constantly, bombard the cell with the reprogramming factors. But from a safety perspective, that is something that the regulators don't like. When you have a foreign genetic material that's persisting in the cell, that introduces carcinogenesis risk, that introduces other sorts of genotoxicity risk. And so, the way that people usually eventually would get rid of the Sendai is just by cell passages. The more you passage the cell, every passage you lose some of the Sendai virus. But even after many, many, many passages, you're still going to have some residual Sendai virus around. And so by using an RNA-based approach, there is no persistent genetic material, and there's no integration risk at all. And so that's kind of the history of why Sendai virus seemed like a great idea at the time and improved reprogramming efficiencies. But as those approaches got in front of regulators, the regulators said, yeah, this is not an ideal way to do this. And so there's just too much safety risk associated with the Sendai virus. So that's what we mean by residual cleanup, is that it stays around for a long time. And the only way to get rid of it is to just pass the cells over and over. And even that does not completely clean the cells of the virus.
Thank you, Scott. That makes sense. Another question is the enhancer included in the cocktail?
Yes, absolutely. So the latest version of our reprogramming cocktail has the enhancer in it. So that's one of the beauties of this kit is it is so easy to use. You don't have to manipulate it in any way. You just dilute some into your culture media and add it to the cells. So yes, the enhancer is included.
Thank you. another question, because culture PBMC is usually needs several growth factors. The question is, what media do we use to culture PBMC?
Okay.
During RNA-LNP transfection, that's different. That's separate question.
So you're asking, do we have a unique media during the introduction of the RNA-LNP? Is that what the question is?
Yes.
So we have cell culture media for PBMC. So Mingjuan, if I state anything wrong, you let me know. Mingjuan is our product management expert. She knows exactly what we sell and don't sell. But I know that we do offer cell culture media for both fibroblasts and PBMCs, and that those have been kind of co-developed along with our RNA-LNP cocktail to give those high efficiency reprogramming results, although we have had clients say that it works well in other culture media as well. Is that accurate, Mingjuan?
I can add on. For as I mentioned earlier, for PBMC, you will need several growth factors, and you have to provide convenience to our customers and to save the time to source different factors from different vendors. We have made a complete PBMC growth media that you can use before the reprogram process. You will need to culture the PBMC for a few days. Yeah, we have the culture media. Regarding the media during reprogramming, there is a commercially available media, so there's readily available. It only needs one. So for that purpose, we provide the media information in our detailed protocol. So the answer to that, we don't have that media, but we provide the information in our protocol. Okay.
And I'm seeing one other question from Jihong Mingjuan that I think you skipped. So Jihong is asking, do we know exactly which cell types in the PBMC are getting reprogrammed into iPSCs? And I don't believe we've done that sort of detailed analysis to figure out if it's lymphoid, erythroid, or myeloid lineage cells that predominantly give rise to iPSCs, Jihong. But we'll ask our technical team, and if they have feedback that, oh yeah, we did look at that, we'll let you know.
Great. Thank you, Scott. Sorry I missed that question.
No worries.
There's another question say that it's a new version of a reprogram kit that works for PBMC and difficult to reprogram kit also works for fibroblasts. So the question is that if I plan to reprogram both fibroblasts and PBMC, which kit can use the same kit for both cell types?
Yeah, so even though we kind of referenced it as the PBMC kit, that kit works well for both PBMCs and fibroblasts.
Thank you, Scott. Yeah, that makes it more convenient for customers who reprogram different cell types. It works for difficult to reprogramming cells, but then it also works for easy to reprogram cells. I have one more question. It said the question is that I understand you offer FDA compliant GMP grade iPSC bank. Do you also provide a research grade iPSC banks?
Yeah, the answer to that is we absolutely are willing to generate banks that are not generated in the GMP environment. So yes, we can set up an NDA and discuss exactly what your project needs are and generate a proposal for you for research grade iPSC banks.
Thank you so much, Scott, for answering those questions. I think it's close to the time for the webinar. Thank you, Scott, for there's another question coming, just came in. Okay. So So the question is, where can I find all the quality control data that has been done on the PBMC reprogramming kit? Scott.
Yeah, most of that information is on our website, Elenia. But we can, if you don't find it, we're going to make a note that you asked this question, and somebody on the team will absolutely send you a link to the appropriate page on the website. And if we need to send you some additional slides, we can do that also. But I'm pretty sure that you'll be able to find it on the website.
Thank you, Scott. Yeah. So I think that we will conclude the webinar. Thank you, Scott, for such an informative presentation. And thank you to everyone who joined in today's webinar. And we truly appreciate your time and engagement. So if you have any additional questions or would like more information, please don't hesitate to reach out to us, either send us email or call us. And as I mentioned earlier, we have a series of the advanced therapy webinars and each month. And please watch your inbox for details about our next session. Have a wonderful rest of your day, and we look forward to seeing you at our next webinar. Thank you.
Thanks, everybody. Have a great day.